Glossary. Acetyl-CoA, acetyl coenzyme A. Small water-soluble molecule that carries acetyl groups in cells. Contains an acetyl group linked to coenzyme A, CoA, by an easily hydrolyzable thioester bond. Acetyl group. Chemical group derived from acetic acid. Acid. In the context of cell biology, an organic molecule that dissociates in water to generate hydronium, H3O+ ions, thereby producing a low pH. Actin filament. Protein filament about 7 nanometers wide, formed from a chain of global actin molecules, a major constituent of the cytoskeleton of all eukaryotic cells and especially abundant in muscle cells. Action potential. Rapid, transient, self-propagating electrical signal in the plasma membrane of a cell such as a neuron or muscle, a nerve impulse. Activated carrier. A small molecule used to carry energy or chemical groups in many different metabolic reactions. Examples include ATP, acetyl-CoA, and NADH. Activation energy. The extra energy that must be acquired by a molecule to undergo a particular chemical reaction. Activator. A protein that binds to a specific regulatory region of DNA to permit transcription of an adjacent gene. Active site. Specialized region of an enzyme surface to which a substrate molecule binds before it undergoes a catalyzed reaction. Active transport. Movement of a molecule across a membrane driven by ATP hydrolysis or another form of metabolic energy. Acyl group. Functional group derived from a carboxylic acid. R represents an alkyl group, such as methyl. Adaptation. Adjustment of sensitivity of a cell or organism following repeated stimulation. Can allow a response even when there is a high background level of stimulation. Adenylyl cyclase. Enzyme that catalyzes the formation of cyclic AMP from ATP. An important component of some intracellular signaling pathways. Adherence junction. Cell junction in which the cytoplasmic phase is attached to actin filaments. ADP, adenosine 5' diphosphate. Nucleotide that is produced by hydrolysis of the terminal phosphate of ATP. See figure 331. Alcohol, organic compound containing a hydroxyl group, OH, bound to a saturated carbon atom, for example, ethyl alcohol. See panel 2.1, pages 64 to 65. Aldehyde, reactive organic compound that contains the HC double bond O group, for example, glyceraldehyde. See panel 2.1, pages 64 to 65. Alkyl group. General term for a group of covalently linked carbon and hydrogen atoms, such as methyl, CH3, or ethyl, CH2CH3 groups, sometimes abbreviated as R. Allele. One of a set of alternative forms of a gene. In a diploid cell, each gene will have two alleles, each occupying the same position, locus, on homologous chromosomes. Allosteric. Describes a protein that can exist in two or more conformations depending on the binding of a molecule, a ligand, at a site other than the catalytic site. Allosteric proteins composed of multiple subunits often display a cooperative response to ligand binding, because the binding of a ligand to one subunit facilitates the binding of ligands to the other subunits. Alpha helix. Common structural motif of proteins in which a linear sequence of amino acids folds into a right-handed helix stabilized by internal hydrogen bonding between backbone atoms. Alternative splicing. Splicing of RNA transcripts from the same gene in different ways, each of which produces a distinct protein. Amide. Molecule containing a carbonyl group linked to an amine. See panel 2.1, pages 64 to 65. Amine. Molecule containing an amino group, NH2. See panel 2.1, pages 64 to 65. Aminoacyl tRNA synthetase. Enzyme that attaches the correct amino acid to a tRNA molecule to form an amino acyl tRNA. Amino acid. Organic molecule containing both an amino group and a carboxyl group. Alpha amino acids, those in which the amino and carboxyl groups are linked to the same carbon atom, serve as the building blocks of proteins. See panel 2.5, pages 72 to 73. Amino acid sequence. The order of amino acid residues in a protein chain, sometimes called the primary structure of a protein. Amino group. Weakly basic functional group, NH2 derived from ammonia. In aqueous solution, an amino group can accept a proton and carry a positive charge. See panel 2.1, pages 64 to 65. Amino terminus. See N-terminus. AMP, adenosine 5' monophosphate. One of the 4 nucleotides in an RNA molecule. AMP is produced by the energetically favorable hydrolysis of ATP. See figure 340. Amphipathic. Having both hydrophobic and hydrophilic regions, as in a phospholipid or a detergent molecule. Anabolism. Reaction pathways by which large molecules are made from smaller ones. Biosynthesis. Anaerobic. Describes a cell, organism, or metabolic process that functions in the absence of air, or more precisely, in the absence of molecular oxygen. Anaphase. Stage of mitosis during which the 2 sets of chromosomes separate and move away from each other. Composed of anaphase A, chromosomes move toward the 2 spindle poles, and anaphase B, spindle poles move apart. Anaphase promoting complex, APC, a protein complex that promotes the destruction of specific proteins by catalyzing their ubiquitination. It is a crucial component of the cell cycle control system. Anion, negatively charged ion, such as Cl- or CH3COO-. Antibody, protein produced by B lymphocytes in response to a foreign molecule or invading organism, binds to the foreign molecule or cell extremely tightly, thereby inactivating it or marking it for destruction. Anticodon, sequence of 3 nucleotides in a transfer RNA molecule that is complementary to the 3 nucleotide codon on a messenger RNA molecule. Each anticodon is matched to a specific amino acid covalently attached elsewhere on the transfer RNA molecule. Antigen, molecule that provokes the production of specific neutralizing antibodies in an immune response. Antiparallel, describes 2 similar structures arranged in opposite orientations, such as the 2 strands of a DNA double helix. Antiport, type of coupled transporter that transports 2 different ions or small molecules across a membrane in opposite directions, either simultaneously or in sequence. APC, see anaphase promoting complex. Apical, describes the tip of a cell, structure, or organ. The apical surface of an epithelial cell is the exposed free surface, opposite to the basal surface. Apoptosis, normal, benign type of programmed cell death in which a cell shrinks, fragments its DNA, and alters its surface so as to activate the cell's phagocytosis by macrophages. Archaea, one of the 2 divisions of prokaryotes, often found in hostile environments such as hot springs or concentrated brine. See also bacteria. Asexual reproduction, any type of reproduction such as budding in Hydra, binary fission in bacteria, or mitotic division in eukaryotic microorganisms that does not involve gamete formation and fusion. It produces an individual genetically identical to the parent. Aster, star-shaped system of microtubules emanating from a centrosome or from a pole of a mitotic spindle. Atom, the smallest particle of an element that still retains its distinctive chemical properties. Atomic weight, mass of an atom expressed in daltons. ATP, adenosine 5' triphosphate. Nucleoside triphosphate composed of adenine, ribose, and 3 phosphate groups that is the principal carrier of chemical energy in cells. The terminal phosphate groups are highly reactive in the sense that their hydrolysis or transfer to another molecule is accompanied by the release of a large amount of free energy. See figures 223-331. ATP synthase, membrane-associated enzyme complex that catalyzes the formation of ATP during oxidative phosphorylation and photosynthesis, found in mitochondria, chloroplasts, and bacteria. Avogadro's number, the number of molecules in a quantity of substance equal to its molecular weight in grams, approximately 6 × 10 to the 23. Axon, long, thin nerve cell process capable of rapidly conducting nerve impulses over long distances so as to deliver signals to other cells. Bacteriodopsin, pigmented protein found in the plasma membrane of a salt-loving bacterium, Halobacterium halobium. It pumps protons out of the cell in response to light. Bacteria, singular bacterium, commonly used name for any prokaryotic organism, but more precisely refers to the eubacteria, the true bacteria, one of the 3 major domains of life. Most are single-celled organisms. Some species of bacteria cause disease. See also archaea. Basal, situated near the base. The basal surface of a cell is opposite the apical surface. Basal body. See centriole. Basal lamina, thin mat of extracellular matrix that separates epithelial sheets and many types of cells such as muscle cells or fat cells from connective tissue, sometimes called a basement membrane. Base, molecule that accepts a proton in solution, also used to refer to the purine or pyrimidines in DNA and RNA, which are organic bases. Base pair, 2 nucleotides in an RNA or a DNA molecule that are specifically paired by hydrogen bonds, for example, G with C and A with T or U. BCL2 family of intracellular proteins that either promote or inhibit apoptosis by regulating the activation of caspases. Beta sheet, folding pattern found in many proteins in which neighboring regions of the polypeptide chain associate side by side with each other through hydrogen bonds to give a rigid, flattened structure. Binding site, region on the surface of a protein, typically a cavity or groove that is complementary in shape to and forms multiple non-covalent bonds with a second molecule, the ligand. Bi-orientation, the symmetrical alignment of sister chromatid pairs on the mitotic spindle, such that one chromatid is attached to one spindle pole and the other chromatid to the opposite pole. Biosynthesis, the formation of complex molecules from simple substances by living cells. Bivalent, a duplicated chromosome paired with its homologous duplicated chromosome at the beginning of meiosis. Bond, see chemical bond. Bond length, the distance between 2 atoms in a molecule, usually those linked by a covalent bond. Bond energy, the strength of the chemical linkage between 2 atoms, measured by the energy in kilocalories needed to break it. Buffer, any weak acid or base that can release or take up protons and thereby serve to maintain the pH under a variety of conditions. C-terminus, carboxyl terminus, the end of a polypeptide chain that carries an unattached carboxylic acid group. Cadherin, a member of a family of proteins that mediate Ca2+ dependent cell-cell adhesion in animal tissues. Calmodulin, CAM, small Ca2+ binding protein that modifies the activity of many target enzymes and membrane transport proteins in response to changes in Ca2+ concentration. Ca2+ calmodulin-dependent protein kinase, CAM kinase, enzyme that phosphorylates target proteins in response to an increase in Ca2+ ion concentration. through its interaction with the Ca2 plus binding protein calmodulin. Calorie, unit of heat. 1 calorie, small c, is the amount of heat needed to raise the temperature of 1 gram of water by 1 degree Celsius. Cancer, disease caused by abnormal and uncontrolled cell division resulting in localized growths or tumors which may spread throughout the body. CAM, C calmodulin. Carbohydrate, general term for sugars and related compounds with the general formula CH2O subscript N. See panel 2-3 pages 68 to 69. Carbohydrate layer, a layer of sugar residues including the polysaccharide portions of proteoglycans and oligosaccharides attached to protein or lipid molecules on the outer surface of a cell. Carbon fixation, process by which green plants incorporate carbon atoms from atmospheric carbon dioxide into sugars, the second stage of photosynthesis. Carbonyl group, pair of atoms consisting of a carbon atom linked to an oxygen atom by a double bond. See panel 2-1, pages 64 to 65. Carboxyl group, carbon atom linked both to an oxygen atom by a double bond and to a hydroxyl group. Molecules containing a carboxyl group are weak carboxylic acids. See panel 2-1, pages 64 to 65. Carboxyl terminus, C terminus. Cascade, C signaling cascade. Caspase, a family of proteases. Members of the family are activated as part of the pathway leading to apoptosis. Catabolism, general term for the enzyme-catalyzed reactions in a cell by which complex molecules are degraded to simpler ones with release of energy. Intermediates in these catabolic reactions are sometimes called catabolites. Catalysis, the acceleration of a chemical reaction due to the presence of a substance, the catalyst, that itself remains unchanged after the reaction. In cells, virtually all biochemical reactions are catalyzed by enzymes to enable them to occur at the temperature of living matter and within the timescale required. Catalyst, substance that accelerates a chemical reaction without itself undergoing a change. Enzymes are protein catalysts. Cation, positively charged ion, such as Na+ or CH3NH3+, pronounced cation. CDK, C-cyclin-dependent kinase. CDK inhibitor protein, protein that inhibits cyclin CDK complexes, primarily to inhibit progress through the G1 and S phases of the cell cycle. cDNA, C complementary DNA. Cell, the basic unit from which living organisms are made, consisting of an aqueous solution of organic molecules enclosed by a membrane. All cells arise from existing cells, usually by a process of division. Cell cortex, specialized layer of cytoplasm on the inner phase of the plasma membrane. In animal cells, it is an actin-rich layer responsible for cell surface movements. Cell cycle, reproductive cycle of the cell, the orderly sequence of events by which a cell duplicates its contents and divides into two.